presentation today. We are Lyxia Corporation, XiaozaoTech Technology, and we are speaking today about better nutrition naturally through microalgae. Microalgae are one of the earliest life forms on Earth. While they are billions of years old, and there are hundreds of thousands of species in existence, only a fraction have been studied. Today we will speak of photoautotrophic algae, which utilize solar energy and photosynthesis to fix carbon from CO2, and do so 10 times more efficiently than terrestrial plants. These microalgae produce the majority of the atmospheric oxygen that we breathe, while consuming what is now known to be a detrimental greenhouse gas. Microalgae form the vital foundation of the food chain, and they are critical to healthy maturation of aquatic predators and prey, and humans alike, being rich in fats, proteins, carbohydrates, antioxidants, and essential vitamins. Many varieties of microalgae are commercially cultivated. Nanochloropsis is a genus of photoautotrophic microalgae comprising 7 known species. They are relatively robust, possess greater than 30% of their weight in lipid content, and are readily commercially cultivated using non-arable land with limited rainfall, sun and seawater, and carbon dioxide with limited additional nutrients. The ability to produce oil from microalgae with just sunlight and CO2 has drawn billions of dollars of investment into biofuel production. This pursuit of biofuel has grown the knowledge base and cultivation techniques. The identification of certain high-value fatty acids within microalgal oil caught the interest of the nutrition industry, which adapted best practices from biofuel producers to nutrition-focused farms. The focus of our farm is the polyunsaturated fatty acid eicosapentaenoic acid, or EPA. Omega-3 polyunsaturated fatty acids are critical to healthy animal and human development, most notably eicosapentaenoic acid, EPA, and docosahexaenoic acid, DHA. EPA supports heart health, blood pressure, the immune system, and mood, while DHA supports brain and eye development and maintenance from the earliest stages of fetal development through to old age. These fatty acids are deemed essential nutrients, as we cannot sufficiently produce them and must obtain them from our diets. The principal source is from fish. Cold water fatty fish, such as salmon, cod, anchovy, herring, and sardine. To best ensure sufficient supply of these essential nutrients, we should eat 2 servings of such fish each week, or take a supplement, such as fish oil. Fish are like us in that they actually don't produce these fatty acids. Big fish eat little fish, who ate smaller fish, who ate smaller guys still, who ate microalgae. Our company is not involved in this fishy process. We directly cultivate microalgae and extract the essential omega-3 in its natural form. Doing so reduces pressure on our planet's fisheries, greens the planet through sustainable algae culture, reduces greenhouse gas emissions to the atmosphere, and creates jobs in regions still emerging economically. At Lyxia XiaozaoTech, we are producing Algolab natural EPA, a better, sustainable source of EPA. Algolab natural EPA is non-GMO, allergen-free, and vegan-vegetarian. This is the most beneficial form of EPA as it is in its natural polar lipid forms. Greater than 50% of the fatty acid content are polar phospholipids and glycolipids. Studies have shown more efficient uptake of polar lipids, equating to greater bioavailability than triglyceride or ethyl ester forms. Fish oil does not provide us this essential omega-3 in its natural polar lipid forms, as fish convert excess polar lipids to triglyceride forms for storage in their tissue. Now let's look at some data comparing polar lipids to non-polar lipids and their relative bioavailability. Suchard et al. Published a study in 2011 in the journal Lipids in Health and Disease, looking at the relative uptake of EPA and DHA when presented as polar lipid, this being a phospholipid from krill oil, or ethyl ester, or reesterified triglyceride. Each group consumed a meal augmented with 1,680 milligrams of one of these omega-3 supplement preparations, and plasma levels were measured at 7 time points over a 3-day period. The data showed differing rates and total levels of omega-3s in the plasma, with krill's polar phospholipids realizing the greatest absorption, followed by RTG, and lastly EE. So, through this study, we see EPA and DHA as phospholipids outperformed non-polar esters as found in common fish oil supplements on bioavailability. In this next study, we will now look at 2 different polar lipid preparations. Kagan et al. In 2013 published their study in the same journal, Lipids in Health and Disease, evaluating relative uptake of omega-3s as presented in algal extract of Nanochloropsis oculata versus krill. 10 healthy males consumed a meal followed by a supplement oil containing 1.5 grams of omega-3s. The 1.5 grams of polar algal oil contained only EPA and as phospholipids and glycolipids. The 1.5 grams from polar krill oil contained 1 gram of EPA and a half a gram of DHA, both as phospholipids. Plasma levels were then measured at 12 time points over 10 hours. On this slide, we see the table on the left is showing the total fatty acid composition of each sample. Note the absence of DHA in the algal oil. The table on the right is a breakdown of the polar lipids in each sample by type. Note that the polar lipid content for the algal oil is just 17%, whereas the krill is near twice that at 33% polar lipids. Here we see graphic representation of Kagan's results, with the closed squares representing the algal oil and the closed diamonds representing the krill. The table on the left shows total uptake of both EPA and DHA. The middle table shows uptake of just EPA, and the table on the right shows uptake of just DHA. The results show algal oil above krill, with higher uptake of both total omegas on the left and EPA alone in the center. However, the table on the right showing uptake of DHA alone shows krill had higher uptake of DHA than algal oil had. Now, if you mind the y-axis, these are much lower levels, but it is curious that we see any DHA for the algal oil, as there is only EPA present in that sample. The author posed several theories for why this may be occurring, but no conclusions were drawn. Kagan summarizes in his results that the fatty acids derived mainly from the meal appeared rapidly in the plasma and peaked about 3 hours after consumption. There were independent increases in the plasma concentration of both EPA and DHA for both algal oil and krill oil. The concentration of EPA was higher with algal oil than krill oil at several time points, as we saw in the graph. The concentration of DHA did not differ significantly between oils at any time point. The maximum concentration of EPA was higher with algal oil, and both the area under the concentration curve and the incremental area under curve for EPA were greater with algal oil. There was no difference between these oils in the area under the curve or the incremental area under the curve for DHA. Having made a case for converting from fish oil to algal oil for our omega-3s and exposing the superior bioavailability of polar lipids in our natural EPA, I would like to share a bit about our production. Our open raceway ponds are formed soil, overlaid with impervious virgin polymer membrane. We operate at depths from 30 to 50 centimeters with active agitation and aeration to introduce both atmospheric gases and CO2. The agitation ensures that all algae get equal time in the sunny, shallower regions. Moderate strength sunlight is best for optimal algal growth. Our seawater is drawn, treated, and tested before use and recycled for reuse. Our CO2 is isolated from flue gas, purified and infused into our ponds. We consume 10 to 20 grams of CO2 for every gram of algal biomass produced, and we release pure oxygen in the process. Over the last 8 years, we have developed many processes and design elements unique to us. Our natural algal strain was selected for robust growth, tolerance of variations in environmental conditions, and high EPA production. The shapes, sizes, and operational parameters of our ponds have been optimized for scale progression. Our dewatering process has been designed to conserve seawater and minimize dry down energy requirements. Our lipid extraction process is high efficiency, high yield, and low energy. And our derivative product processing enables many transformations and substantial concentrating of our EPA. As a green operation, conservation of all inputs and outputs is of critical consideration. Seawater, freshwater, and extraction reagents are recovered, recycled, and reused. All algal lipid fractions we produce are destined to one product or another with no waste. Even our lipid-extracted algal powder is a valuable product rich in protein content with fiber and other nutrients. A quick overview of our cultivation process begins with retrieval of algal seed stock and inoculation. Initially, we are working in artificial seawater and artificial light as we advance to each larger volume vessel. Once flasks in the culture lab reach target density, we inoculate our 100-liter columns using natural seawater and artificial light. Once the columns reach target density, we seed small ponds in our greenhouse. At this point, our algae are now under natural but filtered sunlight. Once these ponds reach target density, we move the culture outdoors and up through successively larger ponds. Once our largest ponds reach target density, the algae are harvested. We isolate the algae from the seawater, leaving a paste that is resuspended in fresh water to desalinate the algae. And once again, the water is removed, leaving an algal paste that is now introduced to our dry down process, which yields a fine, dry, whole algae powder of minimal residual moisture. Our whole algae powder is then processed in a food-grade lipid extraction process, yielding Algolab natural EPA, a thick, green, rich oil, rich in polar lipids, natural pigments such as chlorophyll and beta-carotene, and additional nutrients. Now for a look at some of our products. The image on the left is our whole algae powder. It is comprised of greater than 50% protein and 33% lipid content, of which 5 to 8% is EPA. The EPA content varies by season. The middle image is the product of our food-grade lipid extraction, our Algolab natural EPA, with over 50% polar lipids and 15 to 20% EPA. The photo on the right is our protein-rich, defatted algae powder. On this slide, we again see our polar lipid-rich Algolab natural EPA on the left. Below, I indicate that we will also produce a slightly concentrated version with EPA at 25 to 30%. The middle image is our Algolab SEPA-EE. This is a concentrated algal EPA ethyl ester ranging from 20% to over 70%, which we will introduce in 2021. Below this, I note our Algolab primary ethyl ester. This is a crude ethyl ester of our natural oil with EPA from 18 to 25% and natural pigments. On the right, we see our Algolab SEPA EE powder. This is a vegetarian microencapsulation powder with EPA from 17 to 27%. Like the SEPA EE oil, this powder will not be available until 2021. Because our algae are producing EPA in the absence of DHA, we can offer algal EPA-only oils. We can also combine our EPA with algal DHA from Schizochytrium to formulate custom blends of any EPA to DHA ratio that may be desired. Our EPA-only oil also presents a fantastic starting point for production of ultra-high concentration API material for therapeutic applications. Isolating EPA from DHA in fish oil is a costly and time-intensive process. Algolab EPA is a better, more efficient, and cost-wise source of EPA for API production. The initial products that we will be offering include our Algolab Complete Omega Natural Blends. These blends incorporate our natural EPA polar lipids with algal DHA triglyceride from Schizochytrium. We'll be initially launching 4 blends. 2 blends each offer 350 milligram per gram of total omegas, and 2 offer 370 milligram per gram total omegas. The difference between each is the specific ratio of EPA to DHA. These oils feature roughly 50% of our natural EPA by volume. So these blends are deep, dark green rich oils, rich in polar lipids, natural pigments, antioxidants, and other nutrients, and represent as close to a whole algae supplement as possible. This slide details some additional products we will be launching this year. Our Algolab Natural EPA with MCT provides our EPA as polar lipids and does not contain any DHA. This is available in 750 milligram vegetarian liquid hard caps and as bulk oil in 10 kg and 190 kg drums. Next is our Algolab Complete Omega Natural. As noted on the slide before, there are 4 different blends with EPA as polar lipids and DHA triglyceride. These blends are available in 750 milligram vegetarian liquid hard caps and as bulk oil, and also as microencapsulated vegetarian bulk powders. Regarding our facilities and associated certifications, our upstream cultivation site is 200 acres or 80 hectares and will be online and producing in July of 2020. Our downstream production facility is over 2.5 acres, 1 hectare, and will be online in August of 2020. We currently are in pre-audit phase for our FSSC, ISO 22000, HACCP, and CGMP certifications and expect them completed in Q4 of 2020. For product standards, we received our GROS determination for Algolab Natural EPA for the United States market in May of this year, 2020. For the European market, we have begun our EFSA Novel Foods application and expect completion in 2022. Please note that our Algolab DHA products are both GROS recognized and Novel Foods registered with approval for infant nutrition. For warehousing, sales, and support, we currently have 2 locations. In Asia, we are in Guangxi Province of China, and in North America, we are located in Los Angeles, California. It has been my pleasure to introduce you to our company, our technology, and our products, as well as to give you a bit of information on the advancement of omega-3s from algae and compelling data on EPA as a polar lipid. Are you ready to expand your product portfolio with the greatest products to hit the omega-3 market in years? Now is the time for your qualification and formulation work. To request samples and additional information, just email us at sales@lyxia.com. Thank you for your time today. We look forward to hearing from you.